All right, welcome everybody to Standing for Truth. My name is Donny Budinsky, and I am excited for this video as it is going to be another comprehensive refutation of the critics. And I have a lot to discuss. We have a lot to engage here. In this important video, I will be interacting with arguments from several critics, starting with Dr. Stefan Frello on the topic of Neanderthals. And then I will be answering Dr. Dan Stern Cardinale from Creation Myths. I will be answering his challenge on nested hierarchical patterns in unconstrained sequences of the genome. And I will hopefully, if we have enough time, wrap up the program with a little bit of soteriology, which is the doctrine or study of salvation. And so, in light of the fact that we have a lot to go through, again, this is going to be comprehensive, and it is an important video because it is going to sufficiently answer several major challenges and arguments that have been offered from those in the evolution camp. By evolution, we don't simply mean change over time or changes in allele frequencies in populations over generations. We are talking about large scale change. The theory of common descent that says all life, including strawberries, puppy dogs, and you, if you're watching this and you're a human, the theory of common descent argues that all life, including animals and plants, are related through common ancestry. The separate ancestry model, or the biblical model of ancestry, which is the position that I uphold and defend, you will see throughout this video, and it's not the first time that we've done it here on Standing for Truth Ministries, but you will see that the evidence is best explained and supported by the separate ancestry model, which says that not all life is related through common ancestry, that God created special and he created humans independent from any other form of life. That includes chimpanzees, gorillas, orangutans, dogs, fish, bacteria. They're essentially, according to the separate ancestry model, exists separate ancestries. And so, guys, let's get right into it because I do have to say that I am excited for this, as I am always thrilled to be refuting the critics, because it is important to engage our critics and to make sure that their challenges are being answered sufficiently. And here we go. Argument number one. And so, again, refuting the critics, Neanderthals. Dr. Stefan Frello, a PhD cell biologist, or a PhD plant biologist, I think. I've responded to him many times. I recently put out a technical article responding to, it's going to be a series of articles responding to his challenges. The first article focused on the Y chromosome dissimilarity between humans and the great apes, and demonstrated exactly why the separate ancestry model can best account for the numbers in terms of the Y chromosome. And I am currently working on part 2 of that article series, responding to Dr. Stefan Frello. Part 2 focuses on Neanderthals. And so I did want to give a sneak preview into what I will be offering in terms of a response in written form. And I just find it funny that someone like Stefan Frello, a militant critic of separate ancestry and biblical creation, he keeps putting forth this Neanderthal challenge from their genetics as a challenge that apparently precludes the separate ancestry model or makes it impossible. Okay. And I want to show you that the data is in perfect alignment with the biblical creation model. Okay. And so I just want to go over a couple of the points that he's made here. I'll be responding to all of them in an upcoming article, but right here is his main objection: the differences in Neanderthal genomes when compared to modern humans. He would say fossil sequences in general. Okay. And so he would include Denisovans as well and Heidelbergensis. So fossil sequences or fossil humans. And so he says if you keep insisting in high mutation rate due to living in caves, you have to document that modern humans that lived simultaneously with Neanderthals had a significantly lower tendency to live in caves. You also have to quantify the mutational load due to this lifestyle. If you think they had a higher mutation rate for other reasons, you better come up with something more convincing than a paper talking about a few out of thousands of families with high mutation rates. And I briefly responded to him in a previous video because he had mentioned concerning the mitochondrial DNA, Neanderthals. How do you explain the differences? And I have argued that you can explain largely the differences in Neanderthal genomes when compared to extant humans as being the result of an accelerated rate of mutation accumulation. Okay. So his brief comments here, they work to demonstrate that Stefan Frello and other critics of biblical creation really don't fully understand what we're saying in terms of the topic of ancestry. And so that's why I want to go to the slide here, set the foundation a little bit. I have put out an article and I've talked at great length about mitochondrial DNA phylogenetics. And so you'll notice here, you've got all these branches going right back. This is a human mitochondrial DNA phylogenetic tree. All your branches, which represent DNA differences, okay, or mutations, they go back to three major nodes, L, M, and N, which is fascinating when you consider the biblical creation model, where at creation 6,000 years ago, you start with what? You start with two people, Adam and Eve. They start to have children, their children have children, so on and so forth. Then a couple thousand years later, you reach the flood. And so you have a massive bottleneck. You have your flood bottleneck, and you're left with what? 8 people. Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their 3 wives. Mitochondrial DNA is uniparentally inherited. It is passed down on one side, the mother's side. So it is maternally inherited. And we know, according to scripture, and therefore the biblical creation model, that the earth restarted essentially. Every single human being today can be traced back to 3 reproducing couples. Yes, you have Noah and his wife, okay, but it's unlikely that they had other children. And so you have Shem, Ham, and Japheth and their 3 wives. And isn't it interesting that all these branches on this human mitochondrial DNA phylogenetic tree go back to 3 major groups, 3 major positions, L, M, and N. That is amazing confirmation of the biblical creation model. Now, there are objections, and I put out an article addressing those objections. I've also done a video thoroughly engaging those objections. So we're not going to rehash all of that here. I just want to focus on this objection that deals with Neanderthals. And so you'll see Dr. Nathaniel Jensen from Answers in Genesis, he made this phylogenetic tree. And when you add in the fossil sequences, okay, you can see that the branches extend because there's even more DNA differences or mutations. So when you add in fossil sequences, the evolutionists would argue that you're looking at more time, and that means Neanderthals must be a lot older than extant humans, and this tree must therefore go back further than just 6,000 years. Okay. They would assert that this tree is more, at least the data is more in line with the evolutionary story of human origins. And again, you can see the fossil sequences, but this is easily explained in the creation model if you understand the nature as to the Neanderthal existence, where they lived, how they lived, okay, how they're explained based on the data that we know regarding Neanderthals. And that's what we're going to cover here. Okay. And so, yes, we have Neanderthal DNA. A point to consider before we move forward is the fact that we have any DNA at all from fossil sequences when DNA degrades so rapidly, but yet we have DNA and DNA that's good enough to get some solid information, okay, tells us that this DNA cannot be hundreds of thousands of years old. The fact that we have DNA at all that we can look at and analyze and build a model from tells us that Neanderthals must have lived just thousands of years ago. Okay. And so this is where I want to engage Dr. Stefan Frello's arguments. Okay. And so hypermutation. Neanderthals could have been hypermutating in early generations while becoming highly inbred in later generations. I say that because Neanderthals are different genetically and morphologically. They're adapted to a cold ecoglacial environment, but we do have their genes. We know they were a sophisticated people group. I've gone over all the evidence for why. Just a few. They buried their dead. They were clearly spiritual. They knew how to make and control fire. They knew how to navigate the open waters. They fished for tuna. They made weapons. They did all sorts of things that tell us they were a sophisticated, intelligent people group. And again, we also have their genes in us, which tells us that we interbred, we intermingled, we mated with them, we had children, offspring. Okay. Now, Neanderthals, they are 40% less fit. They're highly inbred. They're runs of homozygosity, these massive stretches of DNA that comprise identical letters. Okay, if you're capital A, capital A, capital B, you're homozygous. There's no DNA diversity there. If you were capital A, lowercase a, capital B, lowercase b, you'd be heterozygous because there would be differences there. Okay, you'd be genetically heterozygous. But in the case of Neanderthals, they were genetically homozygous, highly inbred. They suffered from a high genetic load, which makes sense because they existed in small, isolated populations. They also existed in harsh environmental conditions. Now, we know, according to the biblical creation model, that we have after the flood, okay, we have people intermingling, we have population growth, and eventually we have the Tower of Babel event. So people don't actually spread out entirely until the Tower of Babel event, when God confuses the languages. That could have been 200, 300, 400 years after the flood. Okay. And so that gives us an extended period of time after the flood where you have humans largely isolated as compared to spreading out in all parts of the globe. That doesn't happen until the Babel dispersal. And so people are staying predominantly in one part of the globe. They're not following God's command to move out and be fruitful and multiply in different parts of the planet. But Neanderthals could have been a group. It could have started off as just a small family that ventured away from the main human population and entered less than ideal environments, became isolated. We do know we find them buried in caves. Okay, this is how we know that they're post-flood and not pre-flood, because they are buried in caves intentionally, and they exist in the upper rock layers in post-flood areas. Okay, and so they would have become isolated, and that would have resulted in them becoming extremely inbred. And I'm going to touch on more of that in a little bit, but I do want to focus now on the hypermutation. Okay, so we know that living in caves, this is a fact, and yet we understand that Neanderthals and a lot of these so-called hominins, okay, that evolutionary paleoanthropologists will call pre-humans. But when it comes to Neanderthals, there are many paleoanthropologists in the conventional field that would agree Neanderthals are fully human, simply adapted to cold ecoglacial systems. According to the biological definition of species, we interbred with them. We were capable of producing fertile offspring. Therefore, we're the same species. Okay. And so we understand that they did live in caves. They lived in harsh environments. And we know that living in caves can accelerate mutation accumulation due to several factors. Now, here are several factors. Reduce selection pressure against slightly deleterious mutations. That means these deleterious mutations that are pouring in, they are going to flourish in that population. They're not going to be removed by natural selection. They're going to hang around, and that's not good. Okay, that leads to disease, that leads to rapid and accelerated genetic degeneration. You're going to be exposed to certain chemicals, radiation within the cave environment. This is a huge one. This is why Dr. Stefan Frello's argument demonstrates that he doesn't understand fully the biblical creation model. So he wants to say, if you keep insisting in high mutation rate due to living in caves, you have to document that modern humans that live simultaneously with Neanderthals. The model is saying that either pre-Babel or post-Babel, Neanderthals, starting off as a small family, most likely, okay, they would have become isolated from your main group of humans, which would have led to extant humans that we see today. Okay, so Neanderthals would have been in several groups, several tribes, comprising small populations. They were isolated in less than ideal conditions, away from the main group of humans, okay, which would have led to modern humans today. And we understand from the fossil record that Neanderthals, again, we find them in caves. And so some would have been living or taking shelter in caves. But this is where Stefan Frello and other critics miss the major point. And it is a point that guardians of common descent and critics of young earth creation frequently bring up, which is the fact that the global flood, which would have just ended, consisted of what? Excess radiation, heat, and energy. Okay, this year-long flood would have had a lot of heat, radiation, and energy associated with it. Therefore, right after the flood, especially for those first few hundred years, you'd have radiation rocks. And so if you have these humans, these subgroups of humans like Erectus, like Neanderthalensis, Heidelbergensis, early stage Neanderthal before he became highly inbred could have been Heidelbergensis. Okay, you've also got these islanders like Homo floresiensis, that's Hobbit. Okay, Hobbit was subject to reductive evolution due to living isolated on an island, fully human, not evidence for human evolution. You also have Homo naledi, who lived in caves as well, and we understand that they were subject to reductive evolution. Okay, Homo luzonensis is another one. Anyways, right after the flood, these populations of humans that started off very small branched off So these subgroups, sub branches, they became isolated in areas, okay, where they were subject to the radiation from radiation rocks, the excess radiation from the flood. And so being exposed to certain chemicals, specific chemicals, radiation in areas that they were isolated in and living in, this would have greatly accelerated the mutation rate. Now, I want you guys to have a look at some of these up here. And so, do animals living in caves have a higher mutation rate? Yes, cave animals may have a higher mutation rate than surface dwelling animals. Okay. So, and notice this, mutations in DNA repair and replication proteins can become fixed, which can increase the mutation rate, selective pressures, genetic drift, so on and so forth. So the point is, we have a unique time, okay, Stefan Frello, after the flood for a few hundred years, where you have radiation rocks, which we have today, but the radiation would have been at excess because right after the flood. I mean, we're still feeling the effects of the flood. And so right after the flood, you'd have the highest degree of negative effects. Okay. Which I think partly explains a lot of the anomalous morphological and phenotypic features associated with the so-called pre-humans, which were fully human. So this was a unique time. And Neanderthals, again, were isolated. They branched off from the main group of humans. So they have a separate history. They existed in unique environments. And this all explains the data. Okay, Stefan Frello, he is going to deny it. He is going to reject it because you can give the evolutionary critics, you can give these guys sufficient answers, but that doesn't mean they're going to tap out. Because the question is, are they looking for an answer? Are they remaining ignorant on purpose, willingly ignorant? Okay, we understand that the Bible, especially in Romans 1, tells us that the wrath of God is revealed from the heavens on the ungodly who don't want to worship God, who reject God. Okay, they see his creation, which points to a creator, but they'd rather worship the creature. So we shouldn't be surprised. 2 Peter 2:3 says what? In the last days, there's going to be scoffers denying three things: the creation, the flood, right? The world that then was being overflowed with water that perished, and the coming judgment by fire. And these are the things, to no surprise, that we're debating the evolutionary community with. These apologists of common descent. This is what we are debating. The global flood, which is what they deny, explains a lot of what we see. So yes, the unique conditions immediately post-flood would have resulted in accelerated mutation accumulation, which is why we see longer branches when fossil sequences are added. Notice this. So another thing to consider is damaged DNA repair enzymes. So if you have DNA repair enzymes that work amazingly to fix DNA lesions, DNA breaks, you can experience 1 million DNA lesions in a single cell in a single day. Thank God we have what? 50, 60, 70 trillion cells. Okay. Some people more, some people less. And yet you can experience tens of thousands to 1 million DNA lesions in a single cell in a single day. And so thank God we have these incredibly designed DNA repair enzymes that work to fix a lot of these DNA breaks. Of course, they don't fix them. They don't work perfectly. Okay. They're not flawless, which is why many mutations are passed on in the germ cell line every single generation. But, and here's a chicken and egg problem as well for the evolutionary community. Answer me this. This is a bit of a sideshow, but I want an answer to this. Okay. What came first, evolutionist, naturalist, DNA repair enzymes or DNA? Because it turns out that your DNA repair enzymes, which is found in the DNA, the information for the DNA repair enzymes is coded in the DNA. But you need the DNA repair enzymes to repair DNA, or else DNA can't last. With all the DNA lesions, with all the DNA mistakes that take place in a single day in a single cell, any animal population is going to go extinct in a few generations. You need these DNA repair enzymes for organisms to survive. So again, what came first? The DNA repair enzymes? Well, wait a minute. If your answer is DNA repair enzymes, you need the DNA to code for the DNA repair enzymes. Or did the DNA come first? But wait a minute. You need the DNA repair enzymes for the DNA to survive. All right. Or else it'll be destroyed. It'll be beyond repair. Answer me that, naturalist. There's a line of evidence that precludes atheism and naturalism, naturalistic origins. Anyways, you can have, for many reasons, due to exposure to these chemicals, inbreeding, also being exposed to radiation rocks like Neanderthals were most likely exposed to, you can have damaged DNA repair enzymes. And what that means is you have less of these enzymes to fix these DNA lesions that take place every single day in every single cell. Okay. And notice this. Yes, damaged DNA repair enzymes can significantly, notice it's not just a little bit, Dr. Stefan Frello, can significantly speed up the mutation rate in a cell, as their impaired function leads to an accumulation of unrepaired DNA damage, which can then be miscopied during replication. Remember, a mutation is basically an alteration in the nucleotide sequence of DNA, right? ATCG. So it's an alteration in the nucleotide sequence of DNA. It's akin to a typographical error in a text. All right. Resulting in more mutations being introduced into the genome. So you got Neanderthals. They are living in harsh conditions. They are exposed to radiation rocks right after the flood. So that radiation would be more intense, okay, which is going to affect them negatively, of course. They, as a result, could have damaged DNA repair enzymes. They are exposed to certain chemicals. The fact that they're small as well, they exist in small populations, the fixation rate is going to be faster. Now, if they're inbreeding, they exist in small populations, the biggest effect is going to be inbreeding. You're going to see inbreeding reveals the hidden reservoir of genetic mistakes that have been accumulating. Okay, so they could have started off hypermutating, then they got themselves into a position where they are now highly inbred, and so there's been shifts from heterozygosity to homozygosity, so they exist in a state where more DNA areas or locations in the genome are homozygous. Okay, so your biggest effect at that point is going to be the homozygosity, but the fact that mutations are coming about quickly and they exist in a small isolated population, plus you have different tribes. Okay, so are they intermingling with each other? Is there random mating? These are questions that need to be answered. So that means each tribe having their own unique mutations come about, becoming fixed, okay, plus the inbreeding, plus all the mutations that are accumulating. This is going to change their DNA. In many ways, it's going to change their you're going to see morphological degeneration as well in many instances or many areas. And so this is going to change them in a way where they are different. They diverge genetically and morphologically from extant humans. This is the perfect recipe for explaining Neanderthal genetics. Notice this right here. Is fixation faster in small isolated populations? Yes, fixation of alleles happens. So fixation is basically being stuck in place. We have that allele that reaches 100% frequency in a population, okay? An allele is a genetic variant. Happens much faster in small isolated populations compared to large populations. And again, Neanderthals existed in small populations. They'd be changing. This would increase the rate of change while they are inbred. And very importantly, Do radiation rocks increase mutation rates? Yes, radiation rocks, which contain radioactive elements, can increase mutation rates in organisms living in their vicinity due to the ionizing radiation they emit, which can directly damage DNA and lead to genetic mutations. And again, they can also damage DNA repair enzymes. And so, very easy. This is a very easy answer. It's simple, it's elegant, it's clear, it's based on sound scientific data. Okay. It is scientific, it is empirical. And so the critics can reject it, but it's because they're not really looking to tap out. Are they really looking for an answer? A lot of them are straining at a gnat and swallowing a camel. So you have these humans that branch off, starting off as a small family, you could say, a couple people even, and they expand for a while, they're hypermutating, and eventually they stop expanding in their numbers. They become isolated. They exist in a small population. Now inbreeding is going to be at play here. Okay, but you're going to have faster rates of fixation, and they're existing in harsh conditions. They are subject to the negative effects of radiation rocks, and this alone, okay, can speed up their mutation rate to exactly where we need them to be on our phylogenetic tree here. So as you can see, the branches are longer, but that can all be explained by the factors that I just explained. Okay. And so right here, again, factors include reduced selection pressure against slightly deleterious mutations, exposure to certain chemicals, radiation within the cave environment, or just radiation rocks in general, which would be in abundance in certain areas on the planet right after the flood, and altered DNA repair enzymes due to unique cave conditions. More hypermutation. Radiation rocks, small isolated populations equal what? Faster rates of fixation, therefore change. Okay, your biggest effect is going to be the homozygosity because they're inbred. But again, they would have grown in size. Their history could have started off where they are hypermutating, and then they become isolated. They stop expanding in numbers. And what we see is a lot of inbreeding, but also faster rates of fixation, and therefore what? Change. Also harsh environmental condition. Dr. Rob Carter contributed to this, and he makes some good points that I would like to highlight. Okay. Higher radiation loads due to people living on newly exposed rocks that contain uranium. And again, we've already confirmed right here. Do radiation rocks increase mutation rates? Yes. And guess what? This would have been a unique time, Stefan Frello, after the flood, where you would have an increase in the amount of radiation, an increase in the amount of radiation that's being emitted from these rocks due to the effects and nature of the flood itself. This is an interesting one. A fluctuating magnetic field of the Earth, which would produce pulses of increased cosmic ray activity at the surface and changing radionuclide decay rates, could all affect mutation accumulation rates. Recent work has shown that. And I showed this in a previous program, which is why Stefan Frello responded the way he did. So you'll see this. Specific modern individuals can pass on up to 7-fold more mutations in their children than average. So we've seen real-time evidence. I showed the papers in the last program. And so I'll link that program in the description box so I don't have to go over them again. But Stefan Frello's argument is very weak. Dodge, duck, dip, dive, and dodge. That's what these evolutionists do best. And he scoffs, right? He says, if you think they had a higher mutation rate for other reasons, we've already shown that they did. And because they did in their conditions, unique conditions, easily explains their genetics. You better come up. Look at this. He's assertive, that Stefan Frello. You better come up, because he's very committed. He's invested in the science fiction religious. This is evolutionary philosophy, the science fiction religious idea that he's related to a banana plant. He really believes that, capital B. You better come up with something more convincing than a paper talking about a few out of thousands of families with high mutation rates. You are missing the point, Stefan Frello. Neanderthals and other fossil sequences, they are an exception to the rule. And I show you a paper that gives you an exception to the rule, but it gives you the exact evidence that you're looking for, that you have modern individuals that are hypermutating due to what? Due to radiation. Okay, the paper got into the effects, I believe, of chemotherapy. And so you have modern individuals that can pass up to what? 2, 3, 4, no, 7-fold more mutations to their children than average. So yes, the unique negative effects and conditions that Neanderthals existed in post-flood, they could have branched off prior to the Babel dispersal. They could have branched off after, but became isolated in less than ideal conditions and exposed to radiation rocks. There would have been an intense amount of radiation being emitted, and this would have negatively affected them, and it would have sped up their mutation rates to a degree that we don't really see today, because this was unique. This is a unique people group. Look at, study, examine Neanderthals. They were unique. Okay, very unique, but a very sophisticated people. Notice this. These people are rare. That's the point. Neanderthals are unique. We have unique situations in real time where what? We have hypermutation. So Dr. Rob Carter says, these people are rare. 12 out of 21,879 individuals studied, but they are present. The excess mutation rates were associated with defective DNA repair enzymes, exactly what I was talking about earlier, 2 families, or the exposure to chemotherapy agents prior to becoming a parent, 5 families. Also, 9 of the 12 hypermutating individuals were male. This is real-time evidence backing up this hypothesis, this model, explaining the genetics, the genetic differences of Neanderthals when compared to modern humans. The response from someone like Dr. Stefan Frello is basically, nuh-uh, because he doesn't want to believe, guys. And a lot of times it comes down to a heart issue. You can give them all the evidence in the world, okay? But if they have a heart issue, if they don't want to believe, if they're a scoffer, if they want to worship the creator or the creature over the creator, then no amount of these answers are going to work. These are sufficient answers to challenges that they think preclude separate ancestry. This is the best they have to offer, guys. And quite frankly, they're weak, they're underwhelming. Okay. It takes a little bit of digging into the scientific literature to see that we have good evidence backing up these various hypotheses explaining Neanderthals. Also, 9 of the 12 hypermutating individuals were male, and paternal age was a significant contributing factor. Well, there's another point. You have in the pre-flood world, and for a short time after the flood, you have people, male and females, living hundreds of years old. Okay. Now, of course, with the harsh conditions of the post-flood world and how you'd have war, conquest, survival. Okay. This is the rekindling of culture and civilization. And you'd probably have a lot of tribes battling each other, going to war against each other, a lot of competition. So you could have the potential to live hundreds of years old, but with everything going on, there's probably a lot of death, okay, premature death. So you might not see too many with that potential living to the potential that they have in terms of age. Where in the pre-flood world, you'd have more people, okay, living hundreds of years old. Anyways, for the ones that manage to live longer in the post-flood world, immediate post-flood world, guess what? They're going to accumulate more mutations in their lifetime, and they're going to pass on more. And so you could have just a burst of new mutations in a single generation, where on a phylogenetic tree, it looks like a longer branch, which the evolutionary community is going to interpret as being many generations, even though it's just one. Okay. Evolutionists assume evolution to argue against our model. And that's not a very sophisticated way to argue against a different viewpoint. Even though the burst of excess mutations was not associated with disease of any of the children, this does tell us that extrinsic factors can affect intergenerational mutation accumulation rates. And just as we've seen with Dr. Stefan Frello's so-called response to my Y chromosome article, okay, which we can touch on in a video in the future, but currently I am working on the Neanderthal response, as I've already responded adequately to his points on the Y chromosome. This is what we get from guardians of common descent. We get a lot of storytelling. We get a lot of we get a lot of dodging, ducking, dipping, diving, dodging. We get a lot of, but maybe this. Well, what if that? Just consider this, possibly this, just maybe this. It's a lot of storytelling from the evolutionary community. And so that deals with Dr. Stefan Frello, and it really does deal with his challenge there sufficiently, even though he may never want to admit to that. And so next on our agenda is Dr. Dan Stern Cardinale. Now, I do appreciate our good friend Creation Myth's ability to put forth an intriguing challenge that allows us in the world of biblical creation research to put in the time, the energy, the work, the effort to engage in a sophisticated manner the challenge or challenges. And what this kind of back and forth serves to do is helps us who hold to the biblical model of ancestry. It helps us to really refine our model and refine our arguments. And so for that, I do appreciate Dr. Dan Stern Cardinale in his attempt to falsify the separate ancestry model. And so let's have a look here at what we're dealing with. Here is a summary of Dr. Dan's challenge that he has issued on numerous occasions. He's issued it in our open mic debate series going back over a year ago, possibly even a year and a half ago that we did. It was about every couple of weeks we did an evolution open mic challenge, bring your best evolution, change our minds, refute us wrong. And for 2023 and much of 2024, we engaged in many nights of debate. And so he presented this challenge then. He refined it a little bit from my understanding, and he also presented it in his debate with Dr. Jerry Bergman, a very thought-provoking, a technical exchange that I very much enjoyed. And I appreciate Dr. Dan and Dr. Bergman's time for that debate. And so here is a brief summary of his argument. And so Dan says this essentially: if separate ancestry is true, there should be a lack of nested hierarchies or a lack of a nested hierarchical pattern in unconstrained sequences. These sequences should evolve neutrally as they accumulate mutations over time. Therefore, there should be no or very little hierarchical signature between kinds. Okay, he'll argue that there can be hierarchies within kinds, but not between. And he says this is because mutations would accumulate randomly and scramble any hierarchy. Okay. And so let's go over a few things, a few definitions, few important points related to Dan's argument here. And so when you hear the word constrained, what that means is it's limited in its ability. So you have a sequence in the genome, region of the genome that is limited in its ability to change or alter since it's subject to natural selection, as compared to unconstrained. Okay, this is where his argument is mostly focused on unconstrained sequences of the genomes of biological organisms, unconstrained being regions of the genome that are not subject to selection. So if they're not subject to selection, they will change over time as mutations accumulate. Okay. If you really get down to it, Dr. Dan, okay, he does believe that the majority of the genome is still junk and non-functional. Okay. So he would say that most of those unconstrained sequences would be junk. Okay. Obviously, they're not important enough, according to his model, for selection to preserve over time. Okay. Remember, when they look at differences between organisms, they see those differences as a reflection of time. Now, I will say, just in case Dan's listening or he responds to this, Dan would say, for the sake of this argument and challenge, it doesn't matter if it's functional or not. That's what he would say. Okay. But I'm just saying, bigger picture wise, the evolutionary community, those that hold to the junk DNA paradigm, if they're only going to say that 10 or 15% of the genome's functional, well, that means most of it, according to their viewpoint, would be non-functional. Okay, so they see DNA differences as being a reflection of time, right? So they would look at the DNA differences between humans and chimpanzees as being a reflection of time since their last common ancestor and when that common ancestor diverged into two lineages. That would have been less time. Okay, a shorter amount of time has passed between those two organisms than, let's say, you bring in a mouse, compare humans with mice, compare their genomes, you'll see more DNA differences, which they would argue, according to the model of common descent, reflect more time since divergence. So therefore, humans and chimps share a more recent common ancestor than humans and dogs, or humans and fish, or humans and mice. Okay, mice, though, being within the mammal group, right? Kingdom, phylum, class, order, family, genus, species. And so DNA differences being a reflection of time, and therefore the nested hierarchical patterns that exist in the biological world being a reflection of descent with modification rather than design. Okay. And so it is interesting that Dan would basically say that the function aspect of this debate doesn't really matter. But I do want to point out that just because a region is not conserved does not mean that it's not functional. All right. There are sequences in the genome that we understand are not as important as, let's say, other sequences that are involved in heart function or cellular respiration, right? Like the mitochondrial DNA could be subject to more selection because of its importance. Whereas a lot of your autosomal nuclear DNA, you have regions that are there to tolerate error, more error than other positions in the genome where you get hit with a mutation in a gene that's important for the heart or the brain or respiration. Then guess what? That organism is going to die, and there's not going to be reproduction, because that's what selection is: differential reproduction. Who's reproducing the most? Who's having the most babies? Okay. Another factor, another point to consider is that mutations in these sequences, the unconstrained sequences, may have functional consequences that we're simply not yet aware of, because we are only in the infancy of understanding the DNA language. There's still so much we don't know about DNA function. It would be like, and I'll use a couple analogies, it would be like, or illustrations, if I were to go over to Russia and I was hired to be a proofreader. Well, firstly, I don't speak any Russian. And therefore, if I am reading over a book or a manuscript, I am going to have a very difficult time being able to tell the difference between a mistake and that which is not a mistake, because I need to study more to understand the Russian language better. Or you put a 10-year-old under the hood of a car, they're going to assume I'm sure that everything they're looking at is functional. It looks designed, it looks complicated, it looks complex. But once you start asking them the details of what is there under the hood, they're not going to know because they haven't studied it enough to know. And so if you said just start tearing things out that you don't know what it does, maybe it's not as important as other things, they're going to start removing everything. Okay, because they would also be in the infancy of understanding everything that goes into a car to make a car run. And that's where we are in terms of understanding the DNA language. We do understand a lot, but there is a lot we don't know. Okay. And so that's why there could be a lot about what's going on in unconstrained sequences that we simply just don't understand yet. And a good design analogy for what I'm saying could be this. Okay. My car has many significant parts that without them, the car isn't going to run. Okay. So you can consider those constrained or conserved parts. If I'm driving and all of a sudden my engine jumps into the front of the car, well, guess what? There's not going to be any more driving. And that's because the engine's very important. Okay. Your battery busts. You're not going to get very far without a battery. These things are essential and critical to the function of the vehicle itself. So those need to be maintained. Now, your car also has numerous parts that are not as needed, but yet still important, still purposeful, still functional. Okay. That could be your rear windshield wipers. I don't really use my rear windshield wipers all that often, but that doesn't mean that they're not useful. That doesn't mean that they're not designed. That doesn't mean that they don't serve a purpose. And every now and again, we do use them. Okay. In situations where they become necessary. What about our left and right turn signals? Our directional indicator lights. They're important. They have a use, they have a function, they have a good design, but there's a lot of people that are turning right that don't use the right turn signal, even though they should. Or they're turning left and don't use the left turn signal, even though they should. It has a use. Even somebody who's very irresponsible when they're driving, if they never use the turn signals, that doesn't mean that the turn signals are not important. They're just simply not as important or essential or critical as an engine or a battery. Okay. So although good design, good engineering, they aren't as necessary to the function of the vehicle as an aspect of the vehicle like the brakes, the engine, the radiator. Okay. And also, an interesting point to consider is that parts that we don't use as much could also be more prone to breaking down simply because they're not being used, right? It goes back to that old saying, if you don't use it, you lose it. And so you should be utilizing these features, even though they may not be as important or as essential, in order to make sure that they just simply don't degrade. Okay. And so this, again, is akin to what we see in the genome. And just because a sequence is unconstrained does not mean that it's non-functional. That's an important point. That's looking at the bigger picture. Another thing is, how does Dan know that these differences that he even calls unconstrained are the result of mutations? Because remember, it is the evolutionary community, it's the evolutionary model that explains all DNA differences as being the result of mutations over time. The creationist model explains the majority of autosomal nuclear DNA differences as being the result of created heterozygosity or design diversity. And so differences that you see in unconstrained sequences across kinds could be a reflection of the different functional requirements or the unique functions associated with those creatures. And perhaps those are the ones that would be more dissimilar and creature specific. Okay. Because obviously you have some creatures that look a lot different. And so they're going to have different functions, different features that need to be used and utilized as compared to your conserved regions, which could have more similarities across biological creatures because those are the parts that are more similar anyways, like an engine, right? That's going to be pretty specific. That's going to be pretty generalized to all modes of transportation. And so perhaps these created these kinds, they have specific differences that reflect the uniqueness of the respective creatures. Okay, that's certainly a point worth considering. We can also make testable predictions on that. That's for another video, getting into detail on that. And so one of Dan's main challenges here, here's a slide that he used in his previous debate that you can look over. You can also watch his previous debate. I moderate a lot of debates, and I've kind of made it an unwritten rule that I'm just not a big fan of going through even my own debates. I rarely, if ever, review my own debates. I just let them stand, allow the audience to analyze them, to examine them objectively, and come up with their own decision. Okay, so you can go look at that debate, look at his visuals. Here, Dan has used this argument elsewhere, and so I'm engaging it in my own way, the way that I feel is best. Okay, so let's focus now on his challenge in regards to unconstrained sequences. So as we covered earlier, he will argue that if young earth creation is true, if separate ancestry is true, then there should be no nested hierarchies in unconstrained sequences. He says these sequences should evolve neutrally as they accumulate mutations, and so there should be no hierarchical signature between kinds, and supposedly this is an irrefutable challenge. Okay, firstly, before I go through some of these points here on the slide, even if what Dan is saying is true, we've only been accumulating mutations in neutral regions of genomes for what? 4,500 years since the flood. And the genome is big. Okay, your autosomal nuclear DNA, you got 6 billion letters. You get 3 billion from mom, 3 billion from dad. Okay, we're diploid. And so in 4,500 years, you're not going to accumulate enough DNA differences or enough mutations to scramble any hierarchies that are there. There just simply hasn't been enough time. Okay, God could have created hierarchically. That's what it looks like he did, which makes sense because the Bible says that we're created in the image of God, which means there must be something about us that reflects the divine. And it just so turns out that human engineers design and build things in what? Hierarchical patterns, homologous patterns. They even design interesting transitional like vehicles. Okay, interesting mosaics. And so if God created hierarchically, which the evidence seems to suggest that he did, and it's only been 4,500 years since the flood, mutations accumulating neutrally in a neutral fashion are not going to significantly alter any of these created hierarchical signatures or patterns. Okay, but I will say, if God created hierarchically, what that means is baraminology in general, which is the study of created kinds. It is going to be difficult because if God created the design order hierarchically and designed diversity is true, right? God front loads the original created kinds with pre-existing DNA differences. That means if scientists come an hour after the creation event and they start examining all kinds of creatures, all kinds of the original created kinds, and they do perform genetic sequencing and then they do comparative genomics on these creatures. Since God created hierarchically and with pre-existing DNA differences, but if these scientists didn't know, they came about an hour after, you know what they're going to conclude? Descent with modification, because what are they going to find right after creation? Hierarchical patterns, pre-existing DNA differences that just came about, pre-existing from what? From God's hand. Okay, today we have pre-existing differences. We only let's say we accumulate 100 new mutations per person per generation, but yet we're still accumulating many DNA differences. Well, from what? From past generations. So you go back far enough, and eventually you get to the point of creation where God has front loaded DNA differences into the original created kinds. If you were to do genetic comparisons of these creatures, if you're a naturalist, you're going to conclude what? Again, descent with modification, hierarchical patterns due to evolution, but yet they were just created. Okay. And so this is why baraminology can be difficult, not impossible, but difficult, since we would expect overlap between kinds, even though the kinds are unrelated to each other. So we're not just expecting some real distinct boundary. No, you are going to expect overlap, since we are going to expect various degrees of similarities in genetics, physiology, morphology, and anatomy between creatures like cats, okay, felids, or dogs, canid, or bears, ursidae, okay? If God created a hierarchically, that's what we would expect to find. It are overlaps, and overlaps do not demand ancestry. Here's a few other points. Dan assumes that there are no repeat mutations. He assumes there are no parallel mutations. He assumes there's highly probable mutations arising in different kinds, right? See, I would argue that Dan underappreciates how functional the genome is, which means he underappreciates just how many repeat mutations, parallel mutations, and highly probable mutations there are. Okay. And I got some really excellent feedback from Dr. Rob Carter on some of these points as well. And he provided some feedback through Facebook. So I don't think he minds me giving him a shout out there. And so if the genome is more functional, that means there would be more hotspots. And so Dan wants to assert that mutations are largely probabilistic. But I would argue that mutations might just be more deterministic. And what I mean by that is, if God created hierarchically, if the majority of the genome is functional, okay, if design diversity is true, we also know that environment dictates phenotype in many ways. Environment can dictate change in the genome. We know there's strong evidence for non-random change, okay, adaptive change. And so that would apply to different unrelated creatures existing in similar environments. Okay, so if that's the case, then that could explain exactly what we see in terms of the patterns between kinds. Again, even for sake of argument, not enough time has passed for that many changes to to take place. Okay. Whoops. This is what I get for making this slide quickly. This is going to bug me. Not a fan of typos. So let's fix that right there. There we go. There's evolution in real time. The title just experienced a couple mutations there. Here's the next point I have. Since biological organisms share such a high degree of similarity, we will be able to arrange them in nested hierarchies, and there will be overlap in these hierarchies. So even in true mutations, right, the creationist model explains most DNA differences between and within kinds as being the result of created differences. But even in areas where it's true mutations, all right, if they fall out into hierarchies, that's okay. Because if we understand the existence of repeat mutations, parallel mutations, or even highly probable mutations, or the fact that mutations might lean more deterministic rather than probabilistic, especially if most of the genome's functional. I think a lot of this, a way to differentiate, because I like to talk about frequently how there's a lot of overlap in the creation versus evolution debate. There's a lot of lines of evidence that people put forth to argue for their position that are agnostic to the debate. They're non-discriminatory. They're not really working to advance the ancestry debate because both models can explain the data. For example, homology. Evolutionists would simply argue that you have homologous patterns due to descent with modification. We would argue for homologous patterns due to common design. Okay. And it would be like me saying, well, creation is true because the sky is blue or the earth is round. Dr. Dan would say, well, that's ridiculous. That's absurd because I can also, from the evolutionary model, explain why the sky is blue and the earth is round. Let's say the multiverse was true, DC Marvel style. Okay. The multiverse is true. You have an Earth 2. Let's say on Earth 2, evolution's true. And on Earth 3, creation's true. We just know this for a fact, let's say. Okay, if you were to go to Earth 2 where evolution is true and you were to examine all the evidence, even though evolution is true on Earth 2, a lot of the evidence can still be explained by the creation model. Now, if you go to Earth 3 where creation's true, you can look at a lot of the evidence and the evolution model can even account for it, even if creation's true. That's the point. There's a lot of overlap, okay? But a way to test the models and to discriminate. We're looking for discriminating pieces of evidence would be DNA function. If most of the genome really is non-functional evolutionary leftovers comprising genomic fossils, viral fossils, genetic mistakes, okay, then the evidence might favor, especially in terms of what we're discussing here, a model of common descent. Whereas if the majority of the genome is functional, this would lend credence towards the position that says we have hierarchies due to design rather than descent. It would also account better for what I have here: repeat mutations, parallel mutations, mutations that lean deterministic rather than probabilistic. Okay. We also know that we have an analog to the design world. Again, if we have an analog to the design world, then we have confirmation or verification of the design hypothesis, okay, which suggests that there is something about us that reflects the divine. So here we have damage hotspots in the same way that we have damage hotspots or mutational hotspots in the biological world. Okay. And so notice this, we have an analog in the world of engineering and design. Modes of transportation have damage hotspots. Damage hotspots are areas on different modes of vehicles that experience similar patterns of injury. If you were to go to a car lot that had, let's say, 500 cars, and the lot is organized with SUVs over here, vans over here, sedans over here, hatchbacks in this location, okay, you are going to find, especially in the used world, you are going to find similar patterns of damage, even though you're looking at different vehicles from different companies. Okay, companies out of North America, out of Europe, out of Asia. Okay, let's say you're looking at Toyota, Honda, and then you're looking at Volkswagen, or you're looking at Chrysler, Ford, GM. You're going to find all kinds of patterns in unrelated modes of transportation in terms of the car companies, okay? And notice here, are there damage hotspots in vehicles? We have a perfect analog here to the biological world when comparing to the design world, showing that the biological world is indeed designed, okay? Yes, damage hotspots are areas on a vehicle that are more prone to getting damaged. And we have many positions within the genomes of living organisms that are simply more prone to mutations, that are more prone to alterations, that are more prone to degeneration. And so notice this. You have various areas on a vehicle like door sills, bumpers, wheel arches, headlights, lower body panels. Okay. And they're exposed to different what? They're exposed to different environmental conditions, as in you have various kinds of environmental conditions or factors that are affecting vehicles in the same way, because regardless of where the vehicle is in the globe, okay, the environments, although there are various environmental conditions, they can be the same for various modes of transportation. Exposure to elements. Well, that's the same thing with biological organisms. We're exposed to elements. We live in similar environments. We experience similar mutations. So you can have two unrelated creatures in the biological world simply based on environment, simply based on the nature of their functions. They could experience mutations in the same areas independently, okay, that over enough time, these similar mutations are occurring and accruing, they can mimic a pattern of ancestry, even though they're occurring unrelated and in unrelated creatures, not reflective of ancestry, simply due to damage hotspots, which we have an analog in the design world. Okay, various unrelated modes of transportation experience similar areas of damage due to the similar environments they exist in. Nested hierarchies, patterns by design. And so you have a sedan that would be, these are all designed, and yet you'd have a sedan that shares more with, let's say, a hatchback or an SUV. Okay, a large SUV. Even though they're unrelated, they share more than the sedan would with the truck or with a tractor trailer or with a boat. Okay, so you have a natural nested hierarchy. Human engineers are designing in hierarchical patterns not on purpose. They're not out there saying we need to give the biblical creationists evidence for the creation model and creation predictions. No, just the fact that they're designing vehicles in similar ways for functional and purposeful reasons. Okay, these vehicles are naturally falling out in nested hierarchical patterns in the same way that the biological world falls out naturally in hierarchical patterns. And you can even get hierarchical patterns that are mimicked simply due to mutational or damage hotspots. And I would argue that Dan underappreciates how many of those hotspots that exist, and he underappreciates how functional the genome is. He also underappreciates the occurrence of repeat mutations, parallel mutations, and highly probable mutations. Now, another key point that I do want to touch on has to do with the reality that this challenge is actually more of a challenge to Dan. And so I am going to throw this challenge right back on Dan, and I am going to challenge him. We've engaged this in the past, and I believe his answers are insufficient. If we've been evolving for millions and millions of years, okay, accumulating mutations apparently in neutral regions of the genome for millions and millions of years, why are there hierarchical signatures at all in the evolutionary model? Because again, if these mutations are accruing neutrally over millions of years, they should scramble any genetic level homology, any genetic level hierarchy, if deep time evolution was true. Were nested hierarchical patterns and homologous patterns predicted in the genomes of living organisms before their discovery? That's a genuine question. I'd like an answer to that. Okay. Also, the sequences should be changing to the point where these molecular hierarchies are modeled in the evolutionary model, because we understand, and this is a point of contention, but we do understand that the per generation rate does in many ways approximate the long-term accumulation rate or fixation rate. Okay. Fatuma here says what? Since on average, it will take 4 NE generations for such mutations to reach fixation, about the same number of neutral mutations should be fixed every generation. The rate of fixation mutations is theoretically constant and equals, notice this, equals the neutral mutation rate. Okay. So why are there hierarchical signatures in unconstrained sequences at all if deep time evolution is true? Because if you have all these mutations becoming fixed, that means they're stuck in place. That is species-wide change. That is irreversible and irreparable change. I would say damage, because I would argue that the majority of these mutations are not just neutral or strictly neutral, but nearly neutral, low impact. Okay. Now, Dan is going to say, and he has argued in a recent video that, and this is specific to haploid genomes like the mitochondrial DNA, where you have a lot more selection acting upon the mitochondrial DNA. In a nutshell, because I want to do a full response to his video, and since we're well over an hour now, I'm not going to do that here. And so let me give you a sneak preview just to pre-butt something that he could say. Okay, you have synonymous and non-synonymous mutations. Synonymous mutations don't change the amino acid sequence. Okay, the codons change, but what it reads doesn't change. Non-synonymous mutations, on the other hand, they change the amino acids, and so purifying selection should favor synonymous changes over non-synonymous changes, because the synonymous changes aren't really affecting things in a significant manner. Okay, as they're not changing the amino acids, but non-synonymous ones do. All right, and so the ratio of these kinds of mutations, according to Dr. Dan Stern Cardinale and several papers on the issue, looking at the issue, they're ratioed in a way that suggests that the mitochondrial genome is subject to purifying selection. And apparently it's about 50% or more. Okay. Now, here's the thing, is even with purifying selection and purifying selection on autosomal nuclear DNA, where the evolutionist always goes wrong or misses is the fact that, okay, today fixation is basically zero because we have a massive population, people are spread out over the planet. According to the evolutionary model, this is actually unique. This is new in terms of evolutionary history, because for most of their history, in terms of the human evolutionary story, humans are existing for thousands of years in a small population, in tribes. They haven't really spread out into all parts of the globe yet. And so for most of human history, you have mutation rates, you have fixation rates that are fast, okay? Very fast compared to today. And so what you have is a balancing out effect, because according to the evolutionary model, you have small populations, let's say 10,000, 15,000, maybe 30,000 for thousands of years before spreading out and eventually inventing agriculture and expanding greatly in population and spreading out to all parts of the globe, seizing dominion over the planet. And so even with a lot of purifying selection on the mitochondrial chromosome and some purifying selection in autosomal nuclear DNA. Remember, the mitochondrial DNA is incredibly important. Even the evolutionists would admit that it's all functional for the most part. It's functional in respiration, cellular respiration, providing energy. And so, yeah, even a low impact mutation in a region of the mitochondrial chromosome might affect it to the point where selection has to remove it. Because basically what it looks like we're seeing is you have areas of the mitochondrial DNA that have no mutations. Okay. The ratio suggests that mutations have occurred, but selection is acting on them, removing them, purifying the mitochondrial chromosome in order that we can still have kids and reproduce, because the function is so important, critical, it's so essential, right? Okay. Or you could say, well, these areas are cold. You have cold spots and hot spots, and they simply don't experience mutations. Okay. But let's just say, for sake of argument, you have, you know, and I saw Dr. Zach Hancock said, well, you know, that would minimize or reduce the validity of the genetic degeneration model or the genetic entropy model. Or you could say genetic entropy affects more so your autosomal nuclear DNA rather than a haploid genome like the mitochondrial DNA, in light of its functional nature, how critical it is. Okay. Nonetheless, these are points of discussion set for another video, or just a discussion with proponents of evolution in general. And so, even with a lot of purifying selection in both DNA compartments, whether it's the mitochondrial genome, where you have a lot, or the autosomal nuclear DNA, where you can even give them, okay, some purifying selection, it doesn't matter, because there's still going to be a balancing out effect to a degree where things are balanced out, because the creation model also has a period of time unique in the history of mankind where we have humans after the flood. They restart at 8, but 3 reproducing couples, so three couples, 6 people, and people don't spread out until at least a few hundred years later, when God confuses the languages. So that means you have a smaller population existing in the same area, so isolated for the most part, mutations accruing, but because of the isolation and people not spreading out in the smaller numbers of people, you have faster rates of fixation. So you have rapid fixation for this time period, this window after the flood, right? So between the flood and between the Tower of Babel dispersal, you have this period of time where fixation is fast, very fast. And then people start spreading out to all parts of the globe, and you get rapid and exponential population growth globally. Okay, so now fixation rate basically slows down to 0. But why somebody like Fatuma could say that the per generation rate equals the fixation rate is because when you consider all these factors, things are balanced out. There's a balancing effect. Okay. So I would say that, sure, Dan can have purifying selection, but because what we observe today in terms of human demographics and what humans have done according to the evolutionary model to spread out in all parts of the planet and expand, okay, that is, if you have a needle, that's just the very tip of history. But most of the evolutionary model would say that humans existed in smaller populations, fixation rate would have been faster. Okay, notice this. If a population is small, growing but isolated, this is what we would see after the flood. Okay, a small, I mean, you're starting with 6 people, as in you have 3 reproducing couples, Shem, Ham, Japheth, and their 3 wives. That's very small. Okay, but growing, but also isolated because they don't spread out. Will they have a higher rate of fixation? Notice this. Yes, a small, growing, but isolated population, which is what the creation model has, will likely have a higher rate of fixation due to the increased impact of genetic drift, which is more pronounced in small populations and could lead to the rapid fixation of alleles, even if the population is growing. Okay. The isolation prevents new alleles from entering the population, further accelerating this process. Okay. So the point is, you, after the flood, okay, according to the creation model, you have this unique window of time where fixation is fast. In the evolutionary model, they have many thousands of years where fixation is fast. So even though it's slow to non-existent today, and you have purifying selection, I am arguing that there is a balancing out effect in light of all these details where your per generation rate still approximates, might not be exactly the same. Okay, so where you have your per generation rate could be here, and the difference in terms of the fixation rate could be here. But they want the fixation rate way over here and the per generation rate here, where I'd say they're pretty close because of the balancing out effect based on these factors that I've just explained. Okay. But I do want to, in a follow-up video, go over Dan's video here, at least portions near the end, the most relevant points, and also Forrest Valkai, renegade science teacher, his points on mitochondrial Eve, which are going to directly relate to Dr. Dan's points. Okay. Well, that was hopefully informative for everybody. I knew that I had a large task ahead of me, as these are technical topics. They require detailed rebuttals. They require thorough breakdowns. And so we got to, in an hour and a half, I do have more that I want to touch on, including some topics related to soteriology. But as I've been doing in terms of refuting the critics, I put out a massive debunk not too long ago, engaging Stefan Frello, but also Grayson Hawk. Okay. And in today's program, I think we touched on a lot of important issues here as well with Neanderthals, but also Dr. Dan Stern Cardinale's challenge, which is a fun challenge. And in light of everything that I've explained, I do really want to know, and I want to know in an empirical manner how, in light of the nature of fixation equilibrium, but also just the evolutionary model in general, deep time evolution, these mutations accumulating for hundreds of thousands to millions of years, you would have no doubt a scrambling of the hierarchies that we see exist. That tells me that hierarchical patterns are due to design and not common descent, simply because there has not been enough time to what? Scramble these hierarchical signatures. But if deep time evolution were true, I would not expect, for obvious reasons that we've covered here, I would not expect the level of hierarchical signature that we see in both constrained and unconstrained sequences, especially the unconstrained sequences, because it's the evolutionist that argues that these sequences can change neutrally in light of the accumulation of neutral mutations over time, over lots of time. And so this argument, this specific challenge, both of them, I would say, Neanderthals and the challenge from hierarchies and unconstrained sequences, they are put forth as two of the best so-called challenges by the evolutionary community. And therefore, I wanted to dedicate an adequate amount of time. Okay, these challenges deserve that. Because of that, we are at roughly the hour and a half mark. And so I will continue in a follow-up video. Okay, this is premiering today, the beginning of the week, Monday. And the follow-up video will touch more on the mitochondrial Eve issue, but I will also be diving into some soteriological related issues. Okay, so stay tuned, guys. 2025 is going to be another epic year of engaging the critics and refuting challenges. And so I hope everybody enjoyed this program. Do pass it around because this kind of information is important. It is an important aspect of our ministry that we engage the critics and we answer the best challenges that they have to offer. And so God bless you all. Thank you for tuning in, and we will see you all again very soon. God bless.